Hello, everyone. And so I should just start with the introduction, maybe. So I'm a fourth year PhD student, and my PhD work is focused on investigating the role of Huntington NEAT1 interactions in regulating mitochondrial bioenergetics in Huntington's disease. And so Huntington's disease is an autosomal dominant neurodegenerative disorder that is caused by an expansion of polyglutamine repeat in the Huntington gene. This results in the aberrant Huntington protein formation that causes neurodegeneration. Now, there are no disease modifying therapies available. And one of the reasons is because we really don't know what is the exact function of wild type Huntington protein and how mutant Huntington protein dysfunctions. So, in the past 3 years of my PhD, I found that Huntington protein interacts with NEAT1 in the nucleus. This NEAT1 is a long non coding RNA that forms phase separated organelles in the nucleus called paraspeckles. And the function of this NEAT1 mediated paraspeckles is to regulate gene expression. For example, there is a crosstalk between mitochondria and NEAT1 where stress to mitochondria results in increased number of these NEAT1 mediated paraspeckles in the nucleus. And the way they function is by retaining the RNA and proteins that might lead to apoptosis inside these phase separated organelles to prevent their further translation or interaction with other proteins. So we also know that the mutant Huntington protein causes mitochondrial dysfunction by disrupting mitochondria, which releases mitochondrial nucleic acids in the cytoplasm. And this in turn triggers innate immune response, which is a possible mechanism for neurodegeneration. So this brings me to my hypothesis that Huntington NEAT1 interactions are altered in Huntington disease cells, and this contributes to reduced ability of the cell to deal with mitochondrial stress, causing mitochondrial fragmentation, which leads to mitochondrial RNA release in the cytoplasm and triggering the innate immune response activation. So to test my hypothesis, I have three aims. The first is to investigate the function of Huntington NEAT1 interaction in managing mitochondrial stress. Second is to study the mitochondrial bioenergetics in presence and absence of Huntington NEAT1 and also mitochondrial stressors. And my last aim would be to examine mutant Huntington mediated activation of innate immune response. So briefly, I will be using a wild type and Huntington disease patient-derived cell lines as well as human brain tissue samples. I will test for the levels of NEAT1, ATP metabolism, and other biophysical biochemical assays. I will also determine which innate immune response genes are upregulated in the HD cells compared to the controls. So our lab is like closely engaged with the Huntington Society of Canada, where we organize lab visits and present our work in the most accessible language to patients and their families. We are also part of Huntington's Disease Foundation, where we discuss all the results of ongoing clinical trials and also are updated with the various strategies that target Huntington protein. So from my study, it will give a mechanistic insight into the influence of how wild-type Huntington NEAT1 interactions regulate mitochondria and how this interaction is disrupted in presence of mutant form of Huntington. So the underlying mechanisms at play would open new doors for pharmacological modulation for therapeutic benefit. And with this, I would like to acknowledge my supervisor, lab members, collaborators, and MITO2i for giving me the opportunity to present my work. Thank you.
Thank you, Manisha. Now we'll hear from Yalin Sun from Dr. Susan George's lab in the Department of Pharmacology and Toxicology on her talk on amygdala astrocytes accessibility to delta 9 tetrahydrocannabinol induced mitochondrial oxidative stress in adolescents.
What a mouthful. Okay, well, thank you to MITO2i for this opportunity to talk about amygdala astrocyte susceptibility to delta-9 tetrahydrocannabinol induced mitochondrial oxidative stress in adolescents. So with recent cannabis legalization trends across the world, we're seeing an increased prevalence of adolescent cannabis use, which stands around 44% in Canada. And this is of concern because of the increased risk of psychiatric disorders associated with adolescent cannabis use. Across these disorders, the amygdala exhibits impaired functions in anxiety, depression, and schizophrenia. And the amygdala is still not completely mature during adolescence and is therefore vulnerable to exposure to exogenous drugs. And within the amygdala, the tripartite synapse, which is composed of the pre and post neuronal synaptic components, and the astrocyte, together they tightly regulate neurotransmission to guide anxiety and fear behaviors. And THC, the psychoactive component of cannabis, can come into this network to activate cannabinoid type 1 receptors to elicit its acute and long-term effects and possibly disrupt neurodevelopment. So in our investigation of THC's effects on the developing amygdala, we used three main models. So first was the adolescent non-human primate model of chronic THC administration, after which we performed a top-down unbiased cellular proteomic approach of the amygdala tissue. We then validated these results in a second species of rat. These were adolescent and adult rats chronically exposed to THC. And here we're able to look at age-specific effects and behavioral effects. And finally, in the primary rat astrocyte cultures, we're able to determine the mechanism through which THC elicited these effects. So starting off in the adolescent primate proteomics, a top hit we found in this was glial fibrillary acid protein, or GFAP. This is a cytoskeletal astrocyte marker, which when upregulated, as is here with THC exposure, indicates astrocyte activation. So we validated these results with immunohistochemistry in green, and also another inflammatory marker, complement factor B, in red. So with this, we concluded that adolescent THC exposure induced neuroinflammation in the amygdala. In the adolescent and adult rat, we replicated these results in the adolescents with no comparable results in the adult, indicating an age-specific effect. But here we also thought that there was a link between this inflammatory response and oxidative stress. So here I have 4-hydroxynonyl, or 4-HNE, which is a oxidative stress marker that is upregulated only in adolescents exposed to THC, but not in adults. And furthermore, the level of oxidative stress also correlated to anxiety-like behaviors, such that higher levels of oxidative stress correlated to higher levels of anxiety-like behavior. So with this, we concluded that THC selectively induced oxidative stress in the adolescent amygdala linked to anxiety-like behaviors. And finally, in the primary cultured rat astrocytes, here we determined a mechanism where THC directly activated astrocyte CB1 receptors and intracellular calcium signaling cascades to mediate these inflammatory responses. So in the future, we're also really interested in looking at which specific metabolic pathways were disrupted by THC in the astrocytes. In here, looking at it from a bottom-up approach using metabolomics. And we're also interested to see what effects that this dysregulated astrocyte metabolism would have on neurons. And moving this into in vivo models, we also want to see if we restore these metabolic disruptions, if we're able to restore normal mitochondrial calcium dynamics, and also ultimately improve the behavioral impairments associated with adolescent THC. So for patient engagement in the cannabinoid world, there's kind of a disconnect with adolescents really having a worse mental health outcome compared to their adult counterparts. However, most adolescents don't actually perceive cannabis to be harmful for mental health. So in disseminating this information at youth-oriented programs across U of T, we hope to get this message to the target population. And finally, targeting this dysregulated metabolic crosstalk between astrocytes and neurons may reverse the detrimental long-term effects we see with adolescent cannabis use. And with that, thank you very much to all those that made this project possible. And Thank you to MITO2i.
Thank you, Yalin. Next, we have Pavel Powlowski from Dr. Ruth Ross's and Dr. Anna Andre's lab in the Department of Pharmacology and Toxicology.
Hi, everyone. It's great to be here today, and thank you to the organizers of MITO2i for inviting me to share some of my work. Yep. So today I'll be talking to you about some of the work I'm doing in my PhD studies, looking at the interplay of 2-arachidonyl glycerol producing enzyme, DAG lipase alpha, dopamine, and mitochondrial dysfunction in bipolar disorder. So we've heard some nice background on bipolar disorder this morning, so I won't go into it in detail. However, bipolar disorder is an affective disorder which is characterized by drastic changes in mood, ranging from mania to hypomania and depression. And needless to say, these drastic changes in mood can be very disruptive for the patient's maintenance of their work life, their professional life, and many other aspects of life. However, one physiological change which has been associated with bipolar disorder in a number of studies is reduced mitochondrial function, as you all know, the powerhouse of the cell, in which changes have been observed to the levels of electron transport chain proteins, energetic metabolites, calcium homeostasis, mitochondrial morphology, and more. However, interestingly as well, recent research has demonstrated a role for the endocannabinoid system, the target of drugs such as THC found in cannabis, in altering mitochondrial function, whereby THC activating through the CB1 receptor is able to reduce mitochondrial respiration as well as destabilize complex 1 of the electron transport chain. So we're wondering, can we develop new treatments for bipolar disorder by targeting the endocannabinoid system to alter mitochondrial function? So to do this, we're aiming to employ a number of approaches, starting with genetic analysis to look at how the endocannabinoid system is impacted in BD, moving on to using patient-derived iPSC lines, inducible pluripotent stem cells. I'm working with 3 healthy controls, 3 bipolar disorder samples, and 1 bipolar disorder sample with mitochondrial disease. Using these, I am developing neurons with small molecule protocols, and I aim to look at a number of different outcomes, mitochondrial, electrophysiological, and 2-arachidonyl glycerol activity. So just as a brief primer to the endocannabinoid system, for those of you who are not familiar, there are 2 primary endocannabinoids, anandamide and 2-arachidonyl glycerol. We won't be talking much about anandamide today, but for the purposes of the genetics, which you'll see soon, 2-arachidonyl glycerol is produced by the enzymes DAG lipase alpha and beta and is degraded by MAG lipase as well as ABHD6 and 12. And these 2 endocannabinoids primarily exert their effects through the CB1 and CB2 type receptors. So looking at the table on the left first, starting with the whole genome sequencing data we have in the lab from our 3 healthy controls and 3 BD patient samples, we looked for variants that were common to all 3 BD patients that were not found in any of the healthy controls. And what we found was that there seemed to be quite a few variants that were in the 2AG metabolizing enzymes, particularly the ABHD6, 12, and MAG lipase enzymes. This is interesting, however, this is obviously quite a small sample size. So together with our collaborators at the Mayo Clinic, we went on to look using MAGMA, which is a gene-based analysis in the most recent PGC-BD GWAS study, which is comprised of about 42,000 BD cases and 372,000 controls. And what we found when looking at the same gene set of endocannabinoid genes is that it was only DAG lipase alpha that was nearly significantly involved in BD. However, it doesn't quite reach that threshold of significance, as you can see, when you take BD as a whole and you don't try to subtype anything. However, the original publication did also find that DAG lipase alpha was part of a cluster of genes in the vicinity of this SNP that was significant in BD type 1, but not BD type 2. So there is genetic evidence suggesting that DAG lipase alpha plays a role in bipolar disorder. To that end, I am now in the process of generating dopaminergic neurons from these patient samples, as you can see from this immunofluorescent characterization. And currently, I am looking at the firing rate of these neurons using this multi-electrode array platform. And what we are hoping to do is interrogate their response to different drugs and how that changes the firing rate. But you might also be wondering why dopamine and endocannabinoids. This is not my data. This is data from Dr. Catherine Mielnik, a postdoctoral fellow in Ruth Ross lab. And this shows that increasing 2AG is able to exacerbate hyperdopaminergic phenotypes. So in this case, when we take dopamine transporter knockout mice, which are mice that have very high levels of dopamine in the brain due to the absence of the dopamine transporter, and we give them a MAG lipase inhibitor, so we inhibit the breakdown of 2-AG and increase the concentration, it really increases their locomotor activity, which is one of the hallmarks of high dopaminergic tone. So this indicates to us that potentially targeting the endocannabinoid system might be a good way to target dopaminergic transmission in the brain as well. And so throughout this process, I have been listening to the lived experiences of people with bipolar disorder who consume cannabis about how they perceive their symptoms changing. I think it's very important to note that we should not promote cannabis use in bipolar disorder as it can evidently be detrimental. However, there are quite high rates of cannabis use disorder in bipolar disorder, and I think it is important to listen to these individuals about why they decide to consume cannabis and how they feel that their symptoms are changing. And furthermore, depending on the findings of the study, we hope to be able to give back to the community and advise them on why cannabis use may be detrimental. As some benefits and outcomes, we hope to understand how the endocannabinoid system alters mitochondrial physiology in bipolar disorder to develop new treatments. And furthermore, depending on some of the genetic analysis, we may be able to further stratify bipolar disorder patients and allow for some personalized medicine approaches. And with that, I'd like to thank all the members of both labs, in particular Jay, who's been of great help with the genetic analysis, and I believe he'll be giving a lovely talk next.
Thank you, Pavel, and thank you for introducing Jay. So next we have Jaehyoung Choi from Dr. Anuranda's lab in the Department of Pharmacology and Toxicology.
Thank you for the wonderful introductions. How do I skip this? Sorry, I'm not very technically versed, so. Okay. Amazing. Yep, my name is Jay and I'm a PhD candidate at Dr. Anna Andreas' lab. And I would first like to thank MITO2i for letting me present at this wonderful opportunity. And I have learned so much throughout the entire day. My brief presentation is titled The MITO Gene Risk in Psychiatry and Beyond, Multi-Omics Approach via Deep Learning. And I hope to provide a brief summary of our approaches and work, and hopefully the new era of precision medicine in multi-omics and the use of artificial intelligence for precision psychiatry. So as I just said, a new era for research and medicine has been approaching, and this is due to rapid development of new technology, which has enabled us to investigate multi-omic level biomarker applications for investigating the etiology and searching for biomarker, which has been directly applied for multiple clinical applications. But majority of psychiatric disorders are common but very, very complex. And this has made the utilization of biomarkers, identification of biomarkers, even at the omics level in large samples, an ongoing challenge for the field. And I would like to be not disrespectful, but I have a quote that I brought from a paper published in 2020 by Dr. Murray et al. And it says, clinical psychiatry has lagged behind other fields of healthcare in its use of new technologies and routine clinical data for research. And hopefully in the coming years, biomarkers at multi-omic level together with lifestyle risk factors and clinical characteristics will partake in clinical decision-making, not only, but not limited to diagnostics, prognostics, therapeutic selection, drug development, and also resource allocation. So the figure on the left side is a brief summary of our work. And within the global collaboration we have established, we are creating a MITO gene risk score as an approach for identifying mitochondrial specific genomic risk that's associated with development of psychiatric disorders. So with over 200,000 individual level genotype data, we're applying deep learning techniques to both identify and quantify the disease risk attributable to nuclear encoded mitochondrial genetics. And in parallel, we're with our clinical collaborations, we're validating the clinical utility of a mitogene risk score on disease trajectory, diagnostic, and drug response optimization, while also identifying clinical subtypes based on deep clinical phenotyping along with these scores. Also, in parallel, we are investigating blood-based metabolomic panels to identify and stratify patients not only on the genomic scale or the clinical or demographical scale, but metabolomic signatures together with all of them in patients with like psychiatric disorders. And within the next 2 years, we hope to create and validate this novel framework to bring this to the forefront. So while building this project, we were very, very fortunate to communicate directly with patients and patient advocates to consult our ways forward. While I would like you to rather read these wonderful supporting comments, I believe it's very important to note that patients are in urgent need of individualized and precision medicine to deliver another option that can really speed up the processes to treatment and remission. In summary, our project aims to bring together a new multi-omics approach via mitogene, mitometabolome, and deep clinical subtype through machine learning approaches for one main goal: to develop an applicable mitochondrial dysfunction identification tool in clinical psychiatry and a patient stratification tool to shorten patient suffering. I would have to thank many individuals, so I'll just go through them really quickly. So I would like to thank MITO Canada, MITO2i's, Netramark, CI-Chart, and the Bazooki Group for funding parts of my projects and my degree, and also my wonderful and phenomenal collaborators at Mayo, Fondation Fondamentale, University Paris-Créteil, UK Biobank, Interior Health Studies, and the Metabolic Mind, as well as Psychiatric Genomics Consortium and CAMH, and my advisory committee. And I left my lab out because they're in my heart instead of the slides.
Thank you, Jay. So now to end our brain health segment this afternoon, we have one more student talk from Yejin Kang from Dr. Krista Langt's lab in the Department of Pharmacology and Toxicology.
Hello, everyone. My name is Yejin. I'm a PhD student under Dr. Krista Langto's lab. First of all, thank you, MITO, for this opportunity for me to talk about one of my PhD's talk, and also wonderful day today. I've learned so much. So my project focuses on investigating mitochondrial dysfunctions in vascular malcognitive impairment. So vascular malcognitive cognitive impairment of cerebrovascular origins such as hypertension, hypercholesterolemia, diabetes, and so on. So VMCI is a transitional stage between cognitive decline due to normal aging and dementia, and it is often referred as a pre-dementia stage because it provides a critical windows of opportunity for intervention and potentially altering the trajectory of disease. So therefore, it is very important for us to understand VMCI stage better and identify possible modifiable factors that we can possibly slow down the cognitive decline. So it has been known that OS is linked to both cardiovascular disease and neurological disease involving cognitive decline. And we all know that the mitochondria is closely linked to the oxidative stress. So not only mitochondria is a primary sources of reactive oxygen species, but it is also a immediate target. I know some has been already covered about circulatory cell-free mtDNA. It is a small detectable fragment that is released into the bloodstream due to OS or the cell damage. So it can be a marker of mitochondrial dysfunction. Since mitochondrial dysfunction has been implicated in many brain health, I wanted to know whether peripheral CCF mtDNA can be a potential biomarkers of mitochondrial dysfunctions in VMCI. So this leads to my objectives. In my study, I want to compare the peripheral CCF mtDNA levels in patients with VMCI and compare to the cognitively healthy controls. I also want to investigate the effects of antioxidant treatments on mitochondrial dysfunctions in patients with VMCI. So this study is built in from our 24-week double-blinded placebo-controlled clinical trial. This trial is investigating the effects of antioxidant treatments and acetylcysteine supplements and the cognitive performance in the VMCI populations. So VMCI patients are randomized to other antioxidant group or placebo, and cognitive assessments and blood sample collections are done at the 3 time points. So for my first objective comparing CCF mtDNA levels, I'm going to compare the baseline levels between VMCI and cognitive healthy controls. And for my second objective, I'm looking at longitudinal study, whether the CCF mtDNA levels changes over time in VMCI population receiving the antioxidants treatment. So our groups do multiple strategies for the patient engagement. We consult with patients and care partner throughout the study, and we get some advice for the study design and recruitment strategy. We also consult with the patients and family advisory council from the Sunnybrook Hospital to engage experts by experience. And we also know the importance of the knowledge translation. So we disseminate information and scientific literature in layman terms for patients. And as a part of Sunnybrook Research Institute, we communicate with the general public through various platforms as well, such as website and through social medias. So I hope that result of the study will address a gap and need in the VMCI-related research. I hope that our finding can identify a novel biomarker of disease and can help better understand the biological process in the onset of clinical cognitive decline, and also enable early diagnosis and management. And also, I hope to identify appropriate targets for the interventions, such as antioxidant treatment. So future step will be doing a biomarker analysis and to see whether there will be some interesting results. And with that, I thank all my study participants for, and as well as my supervisor, committee member, my lab member and study team, and also our funding provider, CIHR, Alzheimer's Association, as well as MITO2i for this project. Thank you.